Welcome again, guys. In this video tutorial we'll be talking about Tata box. You probably heard this name previously, but you don't know what it really is. So here we go to talk about Tata box. Now as you see here, it is not something like this is TAT all in capital, and actually this stands for all the nucleotide bases in the DNA. So T means here thymine, A means adenine. So these are the bases of DNA. Okay, so the name Tata box, it means it's not virtually a box. It's actually 4 to 5, actually it lasts for 5 to 7 nucleotide sequence that is found in the DNA promoter region. So if you look at here, in the DNA of eukaryotic cell and also the DNA of archaea, in that case you will see that there are certain regions which is called a promoter. Why it is called a promoter? Because this region helps in promoting DNA replication. They helps in promoting transcription, not replication, transcription mainly. So they helps in transcription of the DNA. That means to produce mRNA from the DNA by the use of RNA polymerase enzyme. Right? So here what we know, the promoter promotes in the transcription process. So in this promoter section, there are different elements in the promoter, core elements and other elements. Now this Tata box is one of the core elements of the promoter. It makes the most of the promoter, and it is very important here. So it is nothing but TATA, AT or AA, whatever that is present in a sequence like this, right? Now promoters are the sections where the RNA polymerase first attaches, and then it scans. When it finds a promoter, then only it can find the start site of the transcription, which is further downstream. This is the start site of the transcription, which is further downstream of the promoter region. Remember, this is downstream. And then this is the section of mRNA that they want to produce, right? So promoter is very important section, because promoter actually helps finding the start site of the transcription region, or start site for the transcription to begin, right? Now in this promoter, this Tata box is extremely conserved throughout the evolutionary history. And if you look at different examples, if you look at different types of organisms, you'll find this something is kind of conserved in this particular section. 7 nucleotide, 8 nucleotide sequence, that is TATA-AT, TATA-AA or something like that. It's not exactly the same, but it's very similar throughout the evolutionary history, and that is called a consensus sequence. It is conserved through evolution, right? And it has a very important task for attaching with the polymerase to start the transcription process. Now this Tata definitely has another very much interacting protein that is called TBP, or Tata binding protein. TBP, Tata binding protein, or Tata box binding protein. Now this Tata box binding protein can interact with this Tata box, which is placed upstream of the actual start site. So if this is the start site, everything here, this is the upstream, this is the downstream, this direction is the downstream. So usually the promoter is found, the Tata box is found 25 nucleotide upstream of the actual transcription start site. Okay, and here this TBP, or Tata binding protein, actually interacts with the Tata box, and then it will bring other enzymes, other factors which are called transcription factors, very important during the transcription in eukaryotes. They'll bring upon these transcription factors, polymerases, other types of enzymes, and it will first unwind the 2 strands of the DNA, because DNA is double stranded. Remember, it will help unwinding the strands of the DNA. It will create a bubble like structure. If I draw it here, it will start to form something like this. This is the region for the Tata box. If you remember, this is the region for Tata box, and here they creates this bubble that's called the transcription bubble that forms here, right, due to the presence of TBP and transcription factors and all these enzymes. Now it is extremely important to look at the structure here, T and A. A and T, if we are filled with A and T, we know AT are attached with each other by 2 hydrogen bonds. And those bonds are much weaker than the bond of GC, which has 3 hydrogen bond, right? So AT, if we have a multiple AT rich sequence, it is very easy for us to unwind them, to separate the strands. That's why it is consisting of consecutive T and A. If it is made with G and C, it will be difficult to separate them, right? It is filled with T and A consecutive sequence, so it's very easy to separate the DNA at this Tata box region, and that is done by the binding of TBP and transcription factors there. It will unwind the DNA. It will first separate the DNA strands, then unwind the DNA, and then start transcribing that DNA into mRNA, and then finally it will be translated into proteins. So that is the role of Tata box. Okay, so if you like this video, please hit the like button, subscribe to my channel, and also share this video to all of your friends in social networking sites. Thank you.